Thank you, Bruce, and thanks to everybody for staying late tonight to this last talk of this symposium where we heard already very important data in the field of follicular lymphoma. And I think I'm quite convinced that the field remains fascinating, and I will try to go over with you some more fascinating data that will come up in the next few months, years, or maybe in the next few days during this meeting. So, while there is a whole field of new agents being developed in lymphoma, and some of them may apply more particularly to patients with follicular lymphoma. This may be a list of maybe 9 items. You don't have to worry, I will keep it short. I will not detail everything, but just maybe focus on a couple of them. First, naked antibodies. We have heard about obinutuzumab, and I should say that beside new anti-CD20 such as obinutuzumab and eventually others, I don't think there is much new things here in naked antibody in the field of follicular lymphoma, even when other targets have been studied. So a few words about antibody-drug conjugates, and I think that's a field that is evolving. Many investigators, many companies have tried to develop this concept of bringing a cytotoxic agent within the tumor cells and by directing it to the cell with an antibody that recognizes a specific antigen. And maybe this list is outdated, and I am sure that a couple of other new antibody-drug conjugates have been developed, but you can see that many antibodies recognize different targets, CD79B, 22, 30, 37, 19, 22, 74, and so on. The compounds are usually microtubule inhibitors, so poisons of the mitotic spindle. Some are focused on distinct subtype of lymphoma, and some have different speed in terms of development. One that has been a little bit more clearly investigated in the field of follicular lymphoma is polatuzumab vedotin, which is an antibody recognizing CD79B, which is coupled with a microtubule inhibitor MMAE. And when the antibody penetrates the cells here, it releases a cytotoxic agent, there is a microtubule disruption, and the cell dies. So this is this kind of process. It's a good target because CD79B is expressed on non-B-cell hematologic malignancies. So the result of polatuzumab vedotin have been presented now and published 2 years ago, and I think they are quite interesting because if you look at the population here of patients with follicular lymphoma, you can see that as a single agent, there were 14 patients investigated. Half of them responded, but there are a few patients actually that were treated with a combination of polatuzumab vedotin plus rituximab, and the response rate was 100%. So where do we go from there? Well, there are some toxicities associated with these ADCs, including some slow release of the cytotoxic agent and some neurological toxicities. Prolonged treatment may not be suitable for patients with follicular lymphoma, and in the field there are many other new agents. But I think we should keep in mind this agent. There are studies that have been investigating, for instance, the combination of BR and BR plus polatuzumab, and I think we have to see whether this data confirms the potential benefit of this combination or whether other combinations currently under investigation will show a pathway for this drug. Bispecific antibodies have been developed, as you know, in ALL, blinatumomab, diffuse large B, very few data in follicular lymphoma. The toxicity associated with this first bispecific antibodies probably preclude its wide use in follicular lymphoma, but we do know that there are other bispecific antibodies being developed, targeting on one arm an antigen on the tumor cell, CD19, CD20, and addressing CD3 on the T cell in order to bring the T cell together to the tumor cell, activate the T cell, and increase cell death. One word about immune checkpoint blockers, which is a field with clearly emerging in all oncology with major changes, probably a revolution in the way we are treating other cancer. Well, follicular lymphoma is probably a very interesting target because the antigens PD-1 on T cells and PD-L1s are expressed in follicular lymphoma, but they may not be expressed exactly where we would like them to be. PD-1 is expressed on a subset of T cells called T follicular helper cells, but PD-L1 is not expressed on tumor cell as a mechanism to escape the T cell response, but on some macrophages from the microenvironment. Well, I mean, there was still rationale to bring this drug into the field of follicular lymphoma. The published results are scarce. Probably the most advanced results were published in JCO last year with one of the anti-PD-1 antibody, nivolumab. And in 10 patients with follicular lymphoma, the response rate was interesting, 40%, but there were only 10% CR. And as you can see here from the spider plots, well, 2 or 3 patients had some response lasting a couple of weeks. However, there were also some transient response. There are many studies that try to evaluate either anti-PD-1 or anti-PD-L1 in the field of follicular lymphoma. And I think we are eager to see whether the combination of this drug with an anti-CD20, with a drug such as IMiDs, will potentially build and engage an immune reactions against the tumor cell that will be more efficient to fight the disease and potentially even eliminate the residual tumor cell. So this brings me to a field which is positioned in between immune-based compound and targeted therapy, which is the field of IMiDs. Well, one of the main IMiDs that have been evaluated in the field of follicular lymphoma and other B-cell malignancies is lenalidomide. And when you see this cartoon, taken from a very nice review by our colleagues John Gribben, Frank Mauschoser, and I think Nathan Fowler, you can think, well, that's a lot of work, and that's a lot of things we have learned. On the other side, when you see that, you may say, well, there are a lot of data coming here, but what's the take-home message? And maybe it's not very clear what's the main effect of this drug in B-cell malignancies. You can see that there are effects on T-cell activation, on NK cells, on malignant B cells, and on microenvironment. But probably the most interesting in vitro data that were generated by using this drug was a demonstration by the group of John Gribben, actually, that, and others, that when you incubate T cell or NK cell in the presence of an anti-CD20 antibody and malignant cells and expose these reactions to lenalidomide, you increase the cytotoxicity of the killer cells. So it's a way to increase the activity of anti-CD20. Is that true clinically? Well, I think we have a couple of data. Some data were generated in relapsed follicular lymphoma, and the ECOG group in the US actually ran this randomized trial evaluating either lenalidomide or lenalidomide plus rituximab. And as you can see, the response rate was increased by the combination of the two agents. The rate of complete response almost tripled. And you can see the PFS curves that were prolonged. So that was interesting. It's not a direct comparison of rituximab versus rituximab plus lenalidomide, but it gives already some hint. We all know that the group of the MD Anderson have published this data in the frontline treatment of follicular lymphoma, and Nathan Fowler has published this data now 3 years ago, which was, again, the combination of lenalidomide plus rituximab, so-called R-square, in the frontline treatment of patients with follicular lymphoma, with a response rate that was over 85, 90% in these patients, and some response lasting a couple of months or years. And I think we'll have an interesting update of this study during this meeting. Some of the response were confirmed by PET, some were confirmed using MRDs, the new tools that were described already by Dr. Rummel. Based on these preliminary data, as well as the data obtained in the relapsed patient, 2 randomized studies are being run. One is a study in the relapse setting, which is augment, and it's R-square versus rituximab alone. One is a study already discussed, which is a relevant study, where R-square followed by R-square maintenance is compared to R-chemo followed by rituximab maintenance. Accrual has been completed more than 1 year ago, and I hope that within a couple of months we'll see the result of this study that may potentially change the landscape of first-line treatment in follicular lymphoma if positive. Well, as you may remember, and as mentioned by Andy Davis, obinutuzumab was designed to increase the cell kill based on the increased ADCC mediated by the antibody. So it was clearly a very good rationale to take the best antibody with a drug that may further increase the cytotoxicity. And that was the rationale to design this study, which is a combination of obinutuzumab and lenalidomide for relapsed and refractory follicular lymphoma, induction, some maintenance with both drugs, and then one single drug here. The phase 1 study was presented at ASH, I think, 1.5 years ago. It determines the dose of lenalidomide being 20 milligrams for induction and then 10 milligrams. We use standard dose of obinutuzumab, and I think that probably tomorrow afternoon you will discover the result of this study, which I found quite interesting. So cell signaling, we do know that within tumor cell, there are a lot of signals that are brought by, first, the B-cell receptor, which is clearly delivering survival signal to the tumor cell, but also by other microenvironment receptors, directed receptors, such as chemokine receptors, also TLRs, chemokine receptors, and eventually even adhesion molecules. And most of these receptors actually signals through different pathways, but key molecules are found in these pathways, which are the PI3 kinase and the subunit delta of the PI3 kinase, not exclusively, but at least one of them, but also the Bruton tyrosine kinase, BTK. So it was logical that the drugs that targeted either the PI3 kinase delta or BTK have been investigated in the field of follicular lymphoma, given the emergence of this drug also in the field of chronic lymphocytic leukemia. So one of the first studies to be published was the activity of idelalisib in patients with relapsed indolent lymphoma. And this was patients that were refractory to both rituximab and an alkylating agent. This was a study that recruited 125 patients, published some time ago in the New England Journal of Medicine. As you may remember, the response rate was fairly interesting, and this is updated result presented by Ajay Gopal at ASH in 2014, and now published in a recent update of this trial, when it shows that more than half of these patients that were again refractory to previous therapy actually responded to idelalisib. And according to the quality of response they achieved, some of the response may last a couple of months, as shown for a few patients that reached a CR, while unfortunately a few patients had, I will say, a response that lasted 8 to 9 months if they didn't have a complete response. Well, it's an effective drug. Many of us are still using this drug. Unfortunately, it brings some toxicity, increased transaminase, diarrhea, and we discovered that infectious toxicity, in particular when the drug is used in combination with either rituximab or bendamustine rituximab, 2 randomized trials were halted, and I think we have to see whether this drug can re-emerge with different combination and maybe different precaution in terms of infectious-associated adverse events. While the other drug, ibrutinib, that has been evaluated, data have been presented last month, 6 months ago at ASH, again by almost the same group of investigators. And it was, again, a population of patients refractory, maybe slightly less refractory than the previous one. And again, single-agent ibrutinib here, but the response rate was only on 25%, also the duration of response and the median PFS was quite similar to the one achieved with idelalisib, and the safety was probably different also based on the toxicity profile of ibrutinib. So we do know that the toxicity between BTK inhibitor and PI3 kinase inhibitor are different. We have atrial fibrillation, we have diarrhea, risk of bleeding with the first one, and diarrhea, elevation of transaminase, infection with the second one. We do know that some compounds are actively developed, actually CC292 not anymore, but GS449, acalabrutinib, copanlisib, which is a mixed inhibitor of PI3 kinase alpha and delta, and which result represented recently at ASCO, some promising result, duvelisib, TGR1202, and others. So this is an emerging field. We have to keep in mind that probably this drug acts not only on tumor cells, but also in cells or in the microenvironment. Whether we'll use them as single agent in refractory patients or in combination as doublets with other agents remains to be defined. And like many other kinase inhibitor, we have to ask whether the duration of treatment is something sustainable, both for the quality of life of the patient, we heard what was said earlier, but also financial. What else do we have? Intracellular trafficking, I think it's too toxic for follicular lymphoma, the selinexor. Venetoclax, BCL2 inhibitor, BCL201 being another one earlier in development. We do know that this drug actually target BCL2, which is overexpressed in the majority of follicular lymphoma tumor cells, and which is probably linked to the translocation 1418, one of the founder events of this disease. Venetoclax actually binds to BCL2, inhibits the activity, and enable pro-apoptotic molecules to get activated and to engage apoptosis in the tumor cells. Well, the results of the phase 1b/2 study of venetoclax have been recently presented and I think published 3 months ago in GCO. And as shown here, 29 patients with follicular lymphoma were treated. The overall response rate is interesting, 38%, but the CR rate is maybe not what we had expected. PR also were found in 24% of the patients. So how this drug will fit into the different possibilities of treatment we have remains to be further defined. One word about epigenetics. We do know that among the founding events of the molecular defects in follicular lymphoma, there are a couple of mutations affecting the epigenetic machinery. One of them is the mutation of EZH2. EZH2 is an enzyme which is brought within a more bigger complex, which is called PCR2, which actually changes the methylation of one histone called H3K27. It was found, and there are very elegant models, mouse model, to demonstrate that it's a key player of the maturation of normal B cell, but also lymphoma cells. And the inhibition of EZH2 was demonstrated to have activity in lymphoma cell lines and in animal models, both actually in models where EZH2 was mutated, a mutation which is found is 20 to 25% of follicular lymphoma, and in patients with wild type, in models with wild type. Well, the result of one of the drugs, which is an oral drug, tazemetostat, were reported already at several meetings, including the ASH Lymphoma Biology meeting. And I think it's the first abstract of the plenary session of this meeting. So I encourage you to look at that and to see if this will be a promising agent, which probably this curve suggests this is a fact. Well, I think I didn't mention in the summary some other agents which are CAR T-cell, and despite the fact that these data here are generated on very few patients, 13 patients, these were highly refractory patients, and investigators at the University of Pennsylvania clearly demonstrated that this may be a very active treatment with long-standing remission for patients. So where do we go from there? Well, it was said we should work on the biology of follicular lymphoma. Yes, this is true, but the biology is very complex. And what we have learned is that there is a high heterogeneity on tumor cells based not only on the different mutations they harbor and how this mutation evolves over time with different way of evolving, but even the clone that you found in a given biopsy are heterogeneous, as we have shown in a recent paper. Moreover, we do know that the microenvironment plays a critical role in these patients, and we have to take that into account. So when we want to develop drugs in patients with follicular lymphoma, we have to think that there are some drugs that may eventually be active in all B-cell lymphomas, especially those acting on the immune response or those some cytotoxic agent. But there are also some drugs that may be active in selected subsets that we haven't identified in follicular lymphoma. So there is still a lot of work to be done. The first one is, what is the goal when you develop new agent? Is that cure? Is it palliation? How do we develop drug combination with commonly used regimen, with other new agents? What's the rational design? What are the toxicity issues? I think it's a really exciting time in terms of clinical trials, but I think it's very promising for patients for which we are aiming to make progress. Thank you for your attention, and happy to answer questions.
It's like Trump, you know.
Okay, no, well, you know that the French and US have strange relationships
like
regarding
our president
handshaking.
and your president just kind of avoiding each other there. All right.
It's great. The bit about Idella was great.
Really?
One thing that has made me curious is follicular lymphoma with its overexpression of BCL2, and we have this splendid venetoclax drug, yet compared with mantle cell with the activity 75%, and CLL where it's 50, 60, 70%, and it's kind of not there in follicular lymphoma. So how do you explain that disparity, what we would expect?
Well, I think we don't have a correct answer at the present time, but I may bring two hypotheses. The first one is the fact that it's a key founder event of follicular lymphoma, the translocation, but there are many other events that will occur during the development of lymphoma. We do know that in half of the population here in this room, we can detect 14/18 positive cells, and hopefully the incidence of follicular lymphoma in this room is very limited, if null. Well, you are a very good doctor. We knew that. So an early event, but maybe when lymphoma develops, the cells are not dependent on overexpression of BCL2, so even if you target it, it doesn't change the survival of this cell. The second thing we have to take into account is that BCL2 is actually quite heavily mutated, and despite the fact that the mutation appeared to spare the BH3 domain where venetoclax binds, there may be questions here in terms of evolution of the tumor and the activity of BCL2 and eventually the activity of venetoclax.
Okay. Several members of the audience have asked, what is the role for auto stem cell transplant in follicular lymphoma? Shall we start with Gilles?
Well, I remember many years ago in the same room discussing autologous stem cell transplant in follicular lymphoma, and that was probably one of the hot debates in Lugano that we all remember, with data presented from different groups. At that time, we tried to investigate autologous stem cell transplant in the front line. The study has shown some PFS benefit, but increased toxicity. It was abandoned in first line. It's an option that should be considered in patients that failed primary therapy, especially in patients with early failure. It's still an active treatment. It has some side effects, an increased fatigue for patients, also probably long-term side effects, but it's clearly one of the treatments that provides a very long PFS if you look at data collected with hundreds of patients, such as the ABMT study. So I think it's an option to be discussed with patients when they failed therapy within, for instance, the first 2 years, the ones that didn't reach EFS 24. But again, it's patients to patients, and we should not say it's necessarily the best. It has never demonstrated in a randomized way that it increased survival. And I do know that in some centers it's commonly discussed with patients, in others less. I think it's open.
The patients who relapse quickly frequently have marrow involvement as well, which makes them not terrific candidates for autotransplant.
Yes, I think you can now with anti-CD20 clean the marrow and collect cells. And it has not really been clear whether collecting cells with a few residual follicular lymphoma cells really hampers the result. I think many studies just ask that there are less than 20% lymphoma involvement, which is something you achieved in the majority of the case with a salvage regimen.
And what's the British approach to autotransplant?
So, having stood on this stage many years ago presenting the long-term follow-up of our autologous practice at Barts and Dana-Farber, we clearly see a significant number of patients who 10 years after having undergone autologous transplantation remain free of disease. And Gilles talked about this wording as not curing follicular lymphoma. I believe that a percentage of patients are cured with autologous transplantation. It needs some patient selection. We know that what we can do now with autologous transplantation and upper limits of age and reduction in toxicity is a much safer procedure than it was when we first started doing this. But I certainly think, particularly for patients who have early progression of disease, it should remain an option on the table. Now, you've just seen a lot of curves of new therapeutics in follicular lymphoma. You notice that hardly any of those curves had any form of a plateau on it. If you look at the long-term follow-up with autografts, there is absolutely a plateau on that. So I certainly think, again, it's a conversation with the patient, but it's something we should certainly discuss.
And in Germany?
So yes, we also consider in patients with high risk, that means early progression of the disease, short remission duration, still good performance status, the role of high-dose treatment and autologous stem cell transplantation. As Andrew Davis has just pointed out, he has seen some long ongoing remissions, and so he may discuss if some of the patients are cured. I would like to make another comment to that issue. We see that long ongoing responses with nearly every kind of treatment. So is it due to a positive patient selection, which we decide to make a transplantation, but also in every study reported, we see on the long end after 10 years, ongoing complete responses, and the patients make this debate, doctor, am I cured? I have a 10-year ongoing complete response, and I see that after chemo, plus rituximab, and also after transplantation in first relapse.
So I would just come back on that and just say, I think we've all been in this game long enough not to use the cure word in follicular lymphoma to patients. We see late recurrences all the time.
Yeah, we'll get to that as the last question here. In the US, the number of patients who undergo auto-transplant, I think you can count on your hands and one of your feet. It's very uncommon, particularly more go to allo, and the others are going to CAR T-cell instead. But you got to my last question, which was the same as one of the audience's question. And I want to go from person to person, starting with Gilles. Forgetting transplant, is follicular lymphoma a curable disease? And if you think not, does it matter given the increased number of new active agents?
I think we heard in the introductions the words of Rosemary and the patient's preoccupation. And what they want is to be cured. They are not really keen on getting a treatment and then another treatment and a third treatment and waiting for the next relapse. So I think we should still aim for cure. It may not be possible in most of the patients, but I believe that the long-term result of the modern frontline treatment, the results that have been presented by Matthias Römel at ASCO, the updates that we'll generate with the PRIMA study, hopefully at ASH, will probably identify a substantial proportion of patients that have not relapsed after frontline treatment. Whether they will relapse in the next 10 years, I don't know. But clearly it shows the ways that we have increased this proportion over time. It was maybe 10, 15% in the old studies without rituximab. It's increased to 30, 40% recently, and we may be able to further increase that. What I believe is that we have new ways on developing immune-based approach in this field, and if we want really to cure this patient, we should really further investigate these tools and see how they will fit in the treatment armamentarium in order to build an effective response and eliminate tumor cells. So I think, yes, it's probably curable, and we should aim for that.
Andy, do you think follicular lymphoma is a curable disease now?
A qualified and cautious yes. I completely agree with what Gilles has said, using optimal immunochemotherapy with maintenance strategies now. But there are also these subgroups, there are these groups of patients who have involved field radiotherapy, where we know 50% of those approximately are going to be long-term free of disease. We see a small number of patients who undergo allogeneic transplantation who are free of disease long-term. We've seen this tail on the curve of patients who've received radioimmunotherapy who very long-term have remained disease-free. So I give a qualified yes to that.
Matthias?
So I won't communicate this word cure with the first conversation with the patient. I will make them hope. When it comes to the question of survival, I will say that they will live for sure much longer than 2 years in an average, what we expect. Of course, it can happen that a patient has to die earlier due to some events, but I give them the hope, and I avoid to address that issue cure. If the patient is going to ask me directly, I will say also cautiously, yes, I have observed some very long ongoing responses without any evidence of the disease, and in these circumstances I may believe that these patients are cured, but I sure don't know it. Also, what I have found in my 10-year update, that patients who achieved a complete response after either treatment, BR or CHOP-R, they are alive with a probability of 80% after 10 years. Just remember, this is a patient group who is 62 years old, and in such a typical patient, that's a good prognosis when you can tell them with a chance of 80% you are alive after 10 years when the patient already is 62 years old. However, everything becomes different if you're going to talk to a patient who is 45 years old. And then this question is much more important and relevant.
We've spent a lot of time looking at the Casula curves and POD24 and how badly patients who progress early do. We haven't focused much on the curve, the upper curve, in terms of those patients who do not progress within 24 months. That upper curve is really fantastic in terms of overall survival. It's not progression-free survival, it's overall survival. And that near matches the age-matched population.
May I also say something?
Please. Into the
You
microphone.
know, what I say to our patients, the lymphoma patients, that they shouldn't stick too much to the statistics. That they should believe in that it's possible that they get cured.